And that amplifier's a little high. Let me turn this down a little. What we're going to do today is we're going to, does that sound better? So what we're going to do today is we're going to start into structure and function of the synapse. We left off last time with Langley's experiments on receptor substance. So pick that idea up today. I'll show you a couple of slides. And we're going to go from ideas of the receptor substance to the demonstration of chemical synaptic transmission. It took, about 10 or 15 years to do this. But there was a change in the way we thought about things with this development of the concept of synaptic transmission being the result of chemical synaptic transmission. And these were experiments done in the late 1910s, early 1920s by a guy named Otto Loewi. And basically, Otto Loewi was interested in the autonomic nervous system, and he showed through his experiments in the autonomic nervous system that there was chemical synaptic transmission. And so that was the first indication that we had really good evidence for synaptic transmission using chemicals. I'll walk our way through his experiments, and then we'll spend some time on synaptic structure and function. It was another, 50 years or so, 40 years before they could use electron microscopes to see synaptic vesicles and active zones and voltage-gated calcium channels. And that gave rise to something known as the snare hypothesis. So what we'll see is we'll talk about the structure of the synapse. We'll put the vesicles into the synapse. We'll talk about the site where the vesicles are lined up in the membrane, called the active zone. We'll see that at the active zone, there's these special channels called voltage-gated calcium channels. Again, these are opened by positive charge. Calcium comes in and causes neurotransmitter release. And then in 2012, the Nobel Prize was awarded for understanding something known as the snare hypothesis. This brings together many of the ideas that we'll set up to this point, but this is a molecular description of how neurotransmitters are released. We'll see that there's a transmembrane protein in the synaptic vesicle called VAMP. Sometimes it's referred to as it's called VAMP. And then the next one is syntaxin, and that's going to be the plasma membrane. And SNAP25 is going to be also in the plasma membrane. These three, in response to calcium, are going to wind together and pull the vesicle into the plasma membrane. Then synaptotagmin, we'll see, is going to be the calcium sensor. So calcium is going to come in through the voltage-gated calcium channels, bind to the synaptotagmin. The binding of the calcium to the synaptotagmin is going to allow the transmembrane protein in the vesicle, VAMP, also known as synaptobrevin, syntaxin, and SNAP25 are all three going to wind together and smush the vesicle into the membrane. And we'll talk about how that happens and why it's an important idea. And then, once we've done that, we've gotten as far as describing how the neurotransmitter is being released, but it doesn't tell us much about what the transmitter does at that point. Now remember that the structure is going to be such that we have a presynaptic element and a postsynaptic element separated by a very narrow gap called a synaptic cleft. The neurotransmitter is going to almost instantaneously, within a matter of a couple of microseconds, pass from where it's released to the postsynaptic membrane. On the postsynaptic membrane, the neurotransmitter is going to bind to ligand-gated ionotropic channels. It's a mouthful, but it's descriptive. The ligand-gated ionotropic channel is a channel which binds the neurotransmitter, and almost immediately after binding the neurotransmitter, the channel opens and ions flow through. In our first iteration of this, we're going to talk about the influx of sodium. And because sodium is coming in, sodium is positively charged, it's going to be depolarization. That means it's an excitatory postsynaptic potential. And EPSP is produced by the opening of these ligand-gated channels. EPSPs in the central nervous system are very small. They're less than a millivolt. And so what we'll see then is for that excitatory postsynaptic potential, which is generated in the input region of a neuron, for that EPSP to affect the trigger zone, much in the way we saw on Friday for generated potentials, these EPSPs have to add together, they have to summate. Each one is far too small to bring the membrane to threshold. And so what we'll see is that these small potentials are going to add together at the trigger zone, supplying enough positive charge to bring the membrane to threshold. An action potential will not be fired unless we get enough of these EPSPs adding together to bring the membrane at the trigger zone to threshold to open the voltage-gated sodium channels. We're going to drill down a little farther into some of the details of the EPSPs, and we're going to talk about something known, we're going to talk about the current, the voltage, and the resistance, and this is going to bring us to something known as Ohm's law. Ohm's law, I should have a delta here. Ohm's law says the change in voltage is equal to the number of ions that move, called current, times the resistance. We're going to simplify this a little bit in our preparation because we're going to just talk about the membrane resistance. But we're going to then talk about the idea of how the membrane has a resistance mostly dependent upon the number of potassium leak channels, as we talked about the other day. And so in membranes which have lots and lots of potassium leak channels, the membrane resistance will be low. It's just as a leaky membrane. And so that means it'll take a lot of voltage to produce a particular it'll take a lot of current to produce a particular depolarization that will then arrive at the trigger zone. And we can talk about whether or not the membrane is leaky or whether it's tight. And this will have important implications for the amplitude of the potentials arriving at the trigger zone. And we'll talk about that, and if there's time, we'll get to inhibition. But let's start with a couple of pictures, summarize where we've been. This first image is just one that summarizes shapes of neurons. And in summarizing the shapes of neurons, I'll remind you that we have talked about these neurons in the discussion on Saturday or Friday. And so we'll see these three general shapes of neurons. The one on the left is this sort of general shape. There's the input region, the conductive region, and the output region. This one is the one we talked about yesterday on Friday, where we had an input region where the more we pulled in the membrane, the larger the generator potential. The larger the generator potential, the greater the probability that we would recruit enough voltage-gated sodium channels to open to initiate an action potential that would propagate to the output region of that neuron. Where we finished on Friday was to talk about Langley's experiments, where he had dissected out an individual muscle fiber. And then he could see where the motor neuron would innervate the muscle fiber. He didn't know what it did. He didn't know why it was there. But he could see it. And then he could take that camel's hair brush with nicotine on it, walk along the membrane, and when he arrived at the neuromuscular synapse, the muscle would contract. That was the idea of the receptor substance. And what he was doing is he was walking along the muscle cell until he reached the synapse where the motor neurons synapsed on the muscle fiber, and he saw a contraction. The next image is one that shows one of these synapses. This in black is the axon terminal of the motor neuron. This is sort of a side-on view, and this is a face-on view. You can imagine what Langley was doing is he was walking along the muscle fiber until he reached just at this point where it was right around the neuromuscular synapse, and the nicotine caused something to happen. There was a receptor substance. It told him that there was, for sure, a receptor substance. But what it didn't tell him is what that receptor substance would do. And it didn't tell him how synaptic transmission would occur. Because simply knowing that nicotine activates something on the muscle fiber doesn't tell you very much about how a neuron communicates with the target. So what we want to do here is we want to talk about some experiments that were done in the late teens and early 1920s by a guy named Otto Loewi. Again, don't remember the name. Otto Loewi was working in Gans in Austria, and he was interested in this idea of whether or not chemical synaptic transmission was how neurons would communicate with their targets. And he spent a lot of time thinking about this. How can I prove that this is the case? And this may be an apocryphal story, but the story is that Otto Loewi went home one night, went to sleep, and at 2 in the morning he woke up. He'd had a dream, and he said, I know how to solve the problem. And he had been working with frogs for a number of years, frog hearts. And he knew that if he picked up a hank of the vagus nerve, that he could stimulate the vagus nerve and change the heartbeat. So let me show you what he did. He could take a beaker, effectively, dissect out a frog heart, leave attached to the frog heart this length of the vagus nerve. Another name for the vagus nerve is cranial nerve 10. These were early days, and so he could pick up the cranial nerve. He could pick up the vagus nerve, which went all the way from the frog's brain down to its heart. He could pick it up on a pair of electrodes, and then he could stimulate. He could pass current between these two wires, and that way he could stimulate the vagus nerve. And when he did that, when he stimulated the vagus nerve, when he stimulated cranial nerve 10, what he found is that the heart rate slowed. We're going to spend some time talking about that when we get to the cardiovascular system. For now, just recognize that he knew that he could stimulate the nerve, and he knew that he'd get a response in the muscle. What he didn't know was how those two communicated. It's back to soups and sparks. Is it an electrical continuity, or is it chemical? And he thought about it and thought about it and thought about it. And apparently, in the middle of the night, he realized the way he could do this was a simple experiment. So he went back to the lab in the middle of the night and started killing frogs. And he set up the same experiment. But he set up two experiments side by side. In one, he set this experiment up where he had the frog heart. He had the vagus nerve. He had the vagus nerve over a wire. He stimulated the vagus nerve and he slowed the heart. His insight was, if it's chemical, then some chemical is likely to get into the bath. So if it's a chemical that's being released by the vagus nerve and affecting the heart rate, it must be something that comes out into the bath. With that logic, he then said, well, if I take another frog heart and I take a little bit of the fluid from this bath and put it into another bath with another frog heart, he could ask the question, did it affect the frog heart? And it did. It slowed the heart rate. So now he's pretty lucky on this, but it turns out what he was seeing is that there was something being released by the vagus nerve that was affecting the heart rate, and that was some sort of soluble substance that he could move from this compartment to this compartment. What he called this in typical German fashion was V-A-G-U-S-S-T-U-F-F, Vagisstoff. It didn't tell him very much about what it was, but at least it was some kind of a chemical. And a good friend of his named Henry Dale, who at the time was working in England, found that Vagisstoff was acetylcholine. That was the first indication of chemical synaptic transmission. And because of the importance of this discovery, because this was the first indication we had a chemical that was being released by a neuron and affecting a target, he was awarded the Nobel Prize in 1936. 1936, Henry Dale and Otto Loewi shared the Nobel Prize. But Otto Loewi was working in Austria. And it turns out Otto Loewi was Jewish. So there's a very sad history here. What happens in 1938, if you know something about European history, is the Anschluss. The Nazis invade Austria. They take over Austria, and because Otto Loewi was Jewish, they said, we'll make you a deal. We'll let you go, but you have to turn over all of your winnings from the Nobel Prize. So they basically went into his bank account, took his money, and put the money into the Nazi Party bank accounts, and released him to let him go to England. So he survived, and he came to the United States later and spent a very good career at New York University, and I think he died in the late mid-60s. But he was a very important player. He and Henry Dale, who described Weierstrass as acetylcholine, were both very important players in development of synaptic transmission and the idea of soups and sparks, and he was a strong soups person, saying this is how neurotransmitters are released. But then, of course, people wondered a lot more about how the synapse functioned and how it worked down at the cellular level. And it was another 50 or 60 years before there was the advent of electron microscopy. Electron microscopy allowed people to look at the nervous system at a level of resolution that had never been seen before. And when they did that, they found something consistently true. Whether they were looking at the neuromuscular synapse that I showed you a picture of a moment ago, or whether they were looking in the central nervous system, what they found is that there was a presynaptic element. And then right next door to the presynaptic element, let me put this in another color, right next door to the presynaptic element was the postsynaptic element. And always, 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 between the two, there was this gap called the synaptic cleft. Sometimes you'll see it known as the synaptic gap. But there was a space. We talked about this earlier. In the central nervous system, this would be surrounded by an extension of the astrocyte. But the next thing they noticed is when they looked inside the axon terminal, on the presynaptic side, in the cytoplasm, they were always viewing clusters, hundreds, perhaps thousands of individual spherical structures, which they termed synaptic vesicles. It took several years of work, but what they found as they looked more and more closely, they found, and I'll go over here and amplify this a little bit, they found that if this is the presynaptic membrane, so this membrane is the same as I'm showing here. They found that in the presynaptic membrane, there were a specialized series of channels, which were called voltage-gated. Calcium channels. Now, to be sure, voltage-gated calcium channels occur in other places too, and we're going to talk about them when we get to the heart. But this is a special set of voltage-gated channels that are found in the membrane at the synapse. Then right next door to those voltage-gated calcium channels, they would see synaptic vesicles clustered right up against the membrane. And these are said to be docked vesicles. Taken together, the vesicles docked at the membrane and the calcium channels themselves are said to be the active zone. So now the sequence of events. The sequence of events is, remember, this is the output region of the neuron. So someplace far, far away, there's an action potential that's initiated. As we talked about on Friday and some on Thursday, that action potential is going to propagate along the axon all the way down to this output region of the neuron. When that action potential arrives at the output region of the neuron, it's going to provide positive charge. Because remember, it's depolarizing the membrane. That positive charge will be recognized by these voltage-gated calcium channels. They're gated. So the calcium gates swing open, and calcium comes in. And at that point, it was recognized that the influx of calcium through the voltage-gated channels would cause the synaptic vesicle to fuse to the plasma membrane. We've seen this process before. This is exocytosis. So at this point, it was recognized that there was the structure of the presynaptic cell. It was recognized it was chock full of vesicles. It was recognized that a handful of the vesicles, a very small fraction, were lined up right along the membrane. These were the docked vesicles. And they were waiting for calcium influx to trigger the synaptic vesicle to fuse with the plasma membrane. The morphologists at the time simply said, well, calcium comes in, some way the calcium is causing the vesicle to fuse with the membrane. Then the physical chemists came along and they said, you've got a problem. It's not that simple. It can't be that simple. Because remember that this is a phospholipid bilayer. And it's going to have, and these are typically going to be charged. And then the synaptic vesicle, too, is going to be a phospholipid bilayer. And it, too, is going to be charged. And as you bring these charges closer and closer and closer together, suddenly you get a repulsion. You get these positive charges on both of the phospholipids will push the membranes apart. So the physical chemist said, something more is going on in here. You've got to have something that overcomes the energy barrier of the vesicle plasma membrane. It took several more years to figure that out. And a number of people working in a number of fields were established something known as the snare hypothesis. I'm going to redraw this with the vesicle and work our way through this idea of the snare hypothesis. And basically what they were doing at the time is they were isolating membranes and trying to sort out which proteins were found on vesicles and which proteins were found on the membrane. And after a lot of experiments, some in yeast, some in cell preparations where they were tracking vesicle movement from compartment to compartment, and some in the central nervous system using isolated structures called synaptosomes, where they had pre- and postsynaptic membranes. They came to realize that regardless of where they were looking, there are a handful of proteins that seem to always be found at these sites where vesicles fused with another membrane. And the question of the repulsion of those charges was largely sorted out by what I'm going to tell you now. Here's the plasma membrane. There's a transmembrane protein here. It goes across the plasma membrane and has an alpha helical region here. The name of this protein is called syntaxin. Then up here on the vesicle, there's another transmembrane protein. I'll put this one in orange. This comes across the membrane, comes out into the cytoplasm. And this one is called VAMP. This is vesicle associated membrane protein. Now you commonly see this referred to by two names. One was synaptobrevin. at least for in here, I'm going to use the term VAMP because I find it more descriptive. It tells us that the protein we're dealing with here is associated with a vesicle-associated membrane protein. Then there's a third character, and I'll put this one in green, and it's not a proper transmembrane protein. It's membrane associated, so it's attached to the membrane, usually by some sort of fatty acid. And it has these two regions of alpha helix. And this one is called SNAP 25. Then finally, the fourth character in the story, I'll put this out here in red, is a transmembrane protein up here. I'm going to draw it like a tag. This is called synapto tagmin. Turns out that after years of study, we've discovered that synaptotagmin is the calcium sensor. And there's some discussion about this, there's some debate about this, but what we think is going on is that the action potential arrives at the terminal. When the action potential arrives at the terminal, the voltage-gated calcium channels open and let calcium come in. That's the first step. Then once the calcium comes in, the calcium, which has come in through those voltage-gated channels, let's put a couple of them down here. These are meant to be the voltage-gated calcium channels. When the calcium comes in, it binds to the synaptotagmin. Then it gets a little hazy. And we're not quite sure what happens here. It's not clear if the synaptotagmin, once it binds calcium, instructs these proteins to wind together, or whether the situation is the synaptotagmin is in the way here. Under normal circumstances, it's not bound to calcium, it's in the way, and it prevents these alpha helices from winding together. It's not clear which of those is true, but what's happening is that these alpha helices now begin to interact with one another. And these alpha helices now wind together. It reminds me a little bit of when you were a kid and you had one of those rubber band driven airplanes and you'd wind the propeller and they get tighter and tighter and tighter. It's as though these proteins are winding tighter and tighter together, and as they wind tighter and tighter together, they're pulling the synaptic vesicle membrane into the plasma membrane. It's this energy, it's the energy of the winding together of the proteins which overcomes the repulsion energy of the phospholipids. It's also probably squeezing out some water that's in there. And so fundamentally what you're doing is you're mechanically pushing this membrane with this membrane to lead to those sort of omega figures we see in the upper panel and exocytosis. That's the concept of the snare hypothesis. And it took a long time to sort out. But to me it was interesting because it required the input of 3 different fields. There was one person named Randy Sheckman who was working on yeast mutants, and he found there were certain proteins in yeast that couldn't secrete properly. And there was a fellow named James Rothman who was interested in vesicle trafficking from the endoplasmic reticulum to the ER and then out to the membrane. And he was finding very similar proteins to the secretion mutants they found in yeast. And then finally, a guy named Tom Sudhoff, working at the University of Texas, found that in the central nervous system, there were a very similar set of proteins. And that by putting these 3 stories together, they recognized that the calcium sensor was synaptotagmin, and that these 3 proteins, syntaxin, SNAP25, and VAMP, would wind together and pull the vesicle membrane and smush it down into the plasma membrane to cause neurotransmitter release. That's the concept of the snare hypothesis. Yes?
So is it like the interaction of the R groups between those
So the question here is, what is it about those alpha helices which allows them to wind together? And it probably is R groups. Those R groups, the R groups are on the alpha helix are interacting with water. And then I don't think it's completely understood how these R groups are now going to interact one with the other. But it's somehow these R groups now are going to wind together to make this knot. Now the truth of the matter is it's also going to take energy to unwind those. And there's certain enzymes in the axon terminals that will unwind these and put them back. One set goes back into the vesicle, the other set goes back into the membrane. But we're not going to get into that. But the fact is, these guys do wind together, and they overcome that energy of repulsion, and that energy has to be taken back out by another set of enzymes. Yes?
So it's overcoming the activation energy of releasing the neurotransmitters into another synapse, because I don't understand, like you're talking about like the two positive charges right there because they're closed. I thought that would be like in endocytosis when it's bringing it in, that the positive charges would be lined. But you're saying
anytime, let's imagine this. Here's the plasma membrane, here's the synaptic vesicle. Anytime this gets close to this, we thought simply by letting the calcium come in, this would get close enough to this and they'd fuse together, where they were being held close enough. But the problem is that there's a phospholipid bilayer here. That's in the vesicle, and these are positively charged. And there's a phospholipid bilayer down here, which is part of the plasma membrane. What's more is there's going to be water in here. And so what we have to do is we have to squeeze out the water, and we have to push together these phospholipids. The process of endocytosis is different. In the process of endocytosis, what you have to do, we do it up here. You have to take the membrane in, and there are certain proteins on the outside of the vesicle called clathrin. And the clathrin will coat the membrane and literally pull that membrane in and cause it to invaginate. Then there's another protein down here which wraps around the neck and breaks it off. And so that's the energy here that you have to use to break the vesicle free of the plasma membrane. Does that answer your question?
yeah, a little bit, yeah.
Okay. Yes. So how do the proteins pull the vesicle in? Okay, we're getting a little off target here, but what they're going to do is they're actually going to form a set of pentagons and hexagons. And if you look at this, it's like a soccer ball. And so you have a series, the clathrins, which are sometimes known as triskelions, and they go like this. These are the triskelions, and these will self-assemble into a lattice work. And that lattice work, as it self-assembles, has enough energy to pull the membrane in. But it doesn't have enough energy to fuse the phospholipids down here as another protein, which is a GTPase, that now fuses those. And so that kind of answers both of the questions, how exocytosis is different from endocytosis. This is the endocytosis story. Yes? What's in the synaptic vesicle? Of course. What's in the synaptic vesicle, go back up here. Where do I have them? So in the synaptic vesicle will be neurotransmitter. So in the Otto Loewi's experiment, this was Wegestoff, later found to be acetylcholine. Most commonly in the central nervous system, at the synapse, the sympathetic system and the heart, it's going to be norepinephrine. And the synapse between the parasympathetic system and the heart, it's going to be acetylcholine. So depending on which synapse you're talking about, you'll have different neurotransmitter. And there are probably, I don't know how many there are, about 8 or 10 of them or so. And they will be packaged in those synaptic vesicles.
So it's the binding of the calcium to the
Tagmin.
that causes all of that to happen.
Because the binding of the calcium to the synaptotagmin causes those alpha helical groups to wind together. When they wind together, that smushes the membrane of the synaptic vesicle into the plasma membrane. So now there's an opening here so that the neurotransmitter can be released to the outside world. And that brings us to the end of the neurotransmitter release aspect. We've gotten the neurotransmitter out of the synaptic vesicle, but now we have to do something with it. And that's the next phase here of beginning to activate the postsynaptic cell using the ligand-gated ionotropic channels. So any questions? Yes?
Is this form of exocytosis for all types of neurons?
This form of exocytosis is for all types of neurons. Yes, all types of neurons use this trick. It doesn't matter whether we're talking about glutamate, choline, talking about pretty much the same trick. Anything else? Okay. Oh, yes.
You said it was all three, the SNAP25, the Synaxin, and the VAMP that wind together?
It's all three of those winding together. Let me show you, see if I can get one of my pictures up here. I have it here. So this image is an electron micrograph taken, it's actually of a frog neuromuscular synapse. And it takes a bit to learn how to see these things, but what we're really looking at, these are the synaptic vesicles. These are spheres in here, the synaptic vesicles. They're full of acetylcholine. Here are two vesicles lined up right along the membrane at the active zone. I think this is probably where the calcium channels are. Calcium then comes in, and the synaptic vesicle, through the snare hypothesis, this vesicle will fuse with the plasma membrane. When it fuses with the plasma membrane, then the 5 or 10,000 molecules of acetylcholine that are located inside the vesicle are released from the cell to fuse across this narrow gap. This gap is no more than 30 to 50 nanometers, so it's tiny. The neurotransmitter diffuses across the gap and now binds to receptors in the postsynaptic cell, and that's where we're headed now. Before we get there, let me show you this sort of colorful picture of the vesicle and the membrane. So again, here are the characters. Here's syntaxin. It's the transmembrane protein in the plasma membrane. Here's VAMP. This is the vesicle-associated membrane protein. It's part of the vesicle membrane. And here then is SNAP25. It's shown here sort of wandering in and out of the membrane because it's not a transmembrane protein. It's a membrane-associated protein. Now, this is the cytoplasm up here in purple, and here's the vesicle. Now, this vesicle now is going to, because of this winding together, is going to be pulled into this plasma membrane. This vesicle is going to fuse with the plasma membrane, and the contents, the lumen of the vesicle now, the contents of the lumen of the vesicle will be spilled out into the extracellular space. That's how that system works. Then, the question, of course, arises, what's going to happen on the postsynaptic side? So on the postsynaptic side, we're now going to begin the process of opening these so-called ligand-gated ionotropic channels. So let's just draw one of these synapses again. Imagine one site. And begin thinking about what happens on the postsynaptic cell. So if this is the postsynaptic membrane here, if the presynaptic membrane is found here, I won't go through all the details again, but when the synaptic vesicle fuses the plasma membrane, the 5 to 10,000 molecules of neurotransmitter that were inside the vesicle are released to the outside world. Then here on the postsynaptic membrane are going to be hundreds of these channels. And these channels have a name. These are, in general, called ligand-gated, ionotropic channels. Well, that's a mouthful. What does it mean? Well, we can walk our way through this and say that the neurotransmitter, and this neurotransmitter could be glutamate, it could be acetylcholine, but it's a neurotransmitter, binds to the ligand-gated ionotropic channel. What it's saying is the neurotransmitter is binding to those receptors. The binding of the neurotransmitter to the receptor changes the conformation of this protein down across the channel and opens a gate. So the neurotransmitter has been released from the presynaptic cell, diffuses across the synaptic cleft, binds to the transmembrane proteins here, which are the ligand-gated channels. The binding of the ligand-gated channels causes a channel to open. When the channel opens, sodium comes in. So it's ligand-gated. That means that's telling us that the gate is opened by the neurotransmitter, the ligand. So ligand simply means to bind or to tie. And so the ligand is the neurotransmitter. It binds to the receptor. The binding of the ligand to the receptor opens a channel which allows ions to move. So it's this name, ligand ionotropic channel. The ions in this case that are coming in are sodium. They're positively charged. So this is going to be excitatory. And we can say that this potential that's produced is an excitatory, postsynaptic potential, which we abbreviate as EPSP, excitatory postsynaptic potential. Now, if we draw this out, I'll do this in the backboard, and put this in context to one of our pineapple neurons. Where up here, we have the trigger zone. And the trigger zone, of course, is characterized by the clustering of the voltage-gated channels. The voltage-gated sodium channels and their counterparts, the voltage-gated potassium channels. Now remember, we said that one of the hallmarks of this input region is that it lacks voltage-gated channels and although it lacks voltage-gated channels, it's chock full of ligand-gated channels. But these ligand-gated channels are not randomly distributed in the membrane. The ligand-gated channels are found at these specific sites, at these synaptic sites, where if the blue boxes here are presynaptic elements, so they will have synaptic vesicles that fuse the plasma membrane. They release neurotransmitter. And then I'll just put these in as a couple of green lines where we have the ligand-gated. The neurotransmitter opens these ligand-gated channels. If we were to put an electrode in here and measure the change, we would see that that depolarization produced by the opening of the ligand-gated channels at the synapse will take the membrane potential from about minus 70 millivolts to maybe minus 69.5 millivolts. This is a depolarization of 0.5 millivolts. Remember where we are at here. If we put an electrode in here, we find that the resting membrane potential is about minus 70 millivolts. And it's stable. But to get these voltage-gated channels to open, we have to get the membrane to about minus 55 millivolts. That's a 15 millivolt excursion. We have to depolarize the membrane by 15 millivolts here. And yet, we're only producing about a half a millivolt change at this site. This tells us that we're going to have about 30 of these must add together to reach threshold at this site. And so what we're saying, in addition, is that there are going to be potassium leak channels all on the membrane. So that this half a millivolt potential, which is generated at this site, by the time it gets to this site, may be no more than, say, 1/10 of a millivolt. So if it only depolarizes by 0.1 millivolts, we're going to need like 150 of these to generate 15 millivolts. These have to add together to reach threshold and fire an action potential. And that's where we'll head next time. Yes?
Single neurotransmitter that binds to